Hi, everyone. My name is Julie Forget. I'm the Senior Director of Safety Assessment at the Altasciences Seattle facility. And I will be presenting today on ICV administration in mice with respect to potential effects from the selected pain management and the injection procedure on animals' behavior post-administration. Gene therapy has shown significant potential for treating a wide array of genetic disorders, specifically those impacting the central nervous system. A notable delivery method is the intracerebroventricular or ICV route, where therapeutics agents are directly administered into the cerebrospinal fluid, or CSF, within the brain's ventricles. Since intrathecal injection in mice is not practical, ICV is the more widely used method of choice. As a standard approach in the industry, ICV injections are performed under anesthesia to minimize pain and discomfort during the procedure. However, post-procedure pain management strategies vary across the scientific community, highlighting the need for defining a more standardized approach. This study aims to identify the most suitable post-procedure pain management method to provide adequate relief while minimizing impact on the animal's behavior. Given that CNS study design often include functional observational battery, FOB, it is essential to ensure that control animals exhibit normal or well-characterized behavior to maintain the integrity of the study outcomes. That study was designed with two groups, a positive control group that was administered buprenorphine ER and regular meloxicam without undergoing surgical procedure. And the second group was the surgical group that underwent the ICV injection. And this second group also received the same pain management as the positive control group. The FOBs were performed at 4 and 24 hours post-dose and included assessment of the behavior, sensory motor functions, and physiological responses. The first observation was that we saw an increased activity level at 4 and 24 hours post-dose in both the ICV and the positive control groups, in both the home cage and open field assessments, suggests a positive effect of the selected analgesic. So in that case was the buprenorphine ER, extended release, and the standard meloxicam. So that was when comparing the general behavior with the pre-dose baseline from the positive control group. The second observation was based on the other assessments that indicated a higher incidence of unusual posture and abnormal gait in the ICV group compared to the positive group at post-dose time points. The slightly greater severity of these observations in the ICV group suggests a potential exacerbation of the condition induced by the ICV injection. So buprenorphine ER was suspected to be the primary contributor to the increased activity levels observed post-dose. Therefore, we conducted an extension of this assessment to refine the analgesic regimen. In this follow-up study, standard buprenorphine, as opposed to the extended release version, was used, and the dose level was reduced from 1 mg per kg to 0.1 mg per kg to minimize its potential impact on the activity levels while still providing an adequate pain relief. Meanwhile, the meloxicam ER was selected instead of the standard meloxicam version, but remained at the same dose level at 4 mg per kg. The benefit of selecting one of the two drugs as an ER formulation is to ensure sustained analgesic coverage while reducing the frequency of dosing and minimizing handling-related stress. So preliminary findings from this modified regimen showed a reduction in the incidence of abnormal behavior within the limited sample tested, with no indication of pain or distress. These results suggest that the revised analgesic protocol may provide effective pain management while minimizing unintended behavior alterations, thereby improving the reliability of behavior assessment in CNS studies. Join me on my poster for a more in-depth conversation on the topic, and I hope to see you in great numbers.